Have you ever wondered what sets living organisms apart from non-living things? It all boils down to the presence of a tiny, yet mighty unit of life, the cell. Every living thing, from the tiniest bacterium to the largest animal, is made up of one or more cells. Cells are not only the building blocks of life, but also the fundamental units of function. They carry out a variety of tasks that keep living organisms alive and thriving. And, did you know that the human body contains trillions of cells, each with a specific function? In fact, it's estimated that the human body contains more than 200 different types of cells. The complexity and diversity of cells are truly fascinating. Now, let's understand how our cell theory was made. So, before the invention of the microscope, scientists had no knowledge of cells. The Dutch scientist Anton van Leeuwenhoek was the first to observe and identify cells in living organisms. Robert Brown later built on Leeuwenhoek's work and discovered the nucleus within the cell. The nucleus, which appears as a pink-colored structure in the center of the cell, was named by Brown. And, in 1838, a botanist from Germany named Matthias Schleiden observed multiple plant species and discovered that various cells make up plant tissues. A British zoologist named Theodore Schwanner examined animal cells the following year in 1839 and found that they possess a thin outer layer known as the cell membrane or plasma membrane. Schwanner also researched plant cells and concluded that plant cells have a unique feature called the cell wall in addition to the cell membrane. After that, Schleiden and Schwanner together formulated the cell theory, which stated that plants and animals are made up of cells and productive cells such as proteins and lipids. But the interesting fact was it did not explain the formation of new cells. At the end, in 1855, Rudolf Virchow conducted further research on the cell theory. He introduced a Latin phrase, omni cellula e cellula, which means that new cells come from pre-existing cells. He modified the cell theory to state that all living things are made up of cells and their products, and cells only come from pre-existing cells. This point became a crucial aspect of the cell theory. So, after knowing what is the theory of cell is, we must learn more about the cell itself, and for that let's move on to the topic of overview of cell. The cell research received a further boost after the invention of an electron microscope, and with the help of electron microscope, scientists were able to observe the structure of the cell. After observing it, they found out that the structure of cell varies in plants and animals. If you look at the plant cell, you see that the plant cell is surrounded by the cell wall on the outside and the cell membrane on the inside. In contrast, animal cells do not possess a cell wall as opposed to plant cells. The outermost boundary of an animal cell is enclosed by a plasma membrane instead of a cell wall. Besides the cell wall and cell membrane, the plant and animal cells have a similar dense structure called the nucleus. Within the nucleus, there are thread-like structures known as chromosomes that contain double-stranded helical DNA. Each chromosome consists of genetic material that stores information and helps to pass it from parents to offspring. Different cells can vary in their shape, size, and activity. For instance, Mycoplasma is the smallest cell, measuring only 0.3 micrometers in length, while bacteria can range from 3 to 5 micrometers in length. The ostrich egg is known as the largest isolated single cell, whereas red blood cells have a diameter of about 7.0 micrometers. The longest cell in the human body is considered to be a nerve cell. The sizes and shapes of cells vary widely, but all of them have different roles to play and work together to keep the body functioning properly. So, cells come in different shapes such as round, polygonal, cuboidal, thread-like, or even irregular. The shape of a cell is determined by the functions it needs to perform. Now students, to make your preparation easy and complete, you should definitely check out the notes on cell, the unit of life. And guess what? You can find all of the chapter notes you need in great detail on EduRev. In certain cells, the nucleus lacks a membrane. These structures, along with other organelles in the cell that are not enclosed in membranes, are referred to as prokaryotic cells. On the other hand, cells that have both a membrane-bound nucleus and membrane-bound organelles are known as eukaryotic cells. Prokaryotic cells are like the ones found in bacteria, and they don't have a nucleus enclosed by a membrane. On the other hand, eukaryotic cells are found in plants and animals and have a well-developed nucleus that is also enclosed by a membrane. And all the cell organelles that are present in eukaryotic cells are also enclosed by a membrane. Have you ever wondered what fills up the space between the cell membrane and the nucleus? That's the cytoplasm, a gel-like substance that is present in all types of cells, both prokaryotic and eukaryotic. Let's take a closer look at this vital component of the cell. Cytoplasm is the semi-fluid matrix present inside both the prokaryotic cell and the eukaryotic cell. The cytoplasm is present inner to the cell membrane, but in case of eukaryotic cell, the cytoplasm is not suspended inside the nucleus because it is membrane-bound, but instead of cytoplasm, the fluid nucleoplasm is present inside the nucleus. So, all the content of the nucleus are suspended inside the nucleoplasm, but in the case of prokaryotic cell, all the contents including the nucleus and other cell organelles are suspended inside the cytoplasm. So the cytoplasm present inside the cell is the hub of various activities and chemical reactions. The cytoplasm in a cell hosts many chemical reactions and activities, such as protein synthesis, which helps the cell survive. Inside the cytoplasm are cell organelles, which are structures that perform specific functions. Eukaryotic cells are more advanced than prokaryotic cells and have well-developed, membrane-bound cell organelles such as the endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. Now, let's learn about some of the important eukaryotic organelles. Electron microscopes have allowed us to see tiny tube-like structures scattered throughout the cytoplasm of eukaryotic cells, forming a network called the endoplasmic reticulum, ER. The ER divides the space inside the cell into two compartments, luminal, inside the ER, and extraluminal, in the cytoplasm. Sometimes, ribosomes, for now imaging ribosomes as some small bodies that are found attached to the outer surface of the ER, which is then called rough endoplasmic reticulum, RER. In the absence of ribosomes, it's called smooth endoplasmic reticulum, SER. RER is common in cells involved in protein synthesis and secretion, and is connected to the outer membrane of the nucleus. The SER is responsible for lipid synthesis, and in animal cells, it plays a role in synthesizing steroidal hormones. Let's now move on to one of the most important organelles found in eukaryotic cells, the Golgi apparatus. The Golgi apparatus, also known as the Golgi complex or Golgi body, is a membrane-bound organelle found in eukaryotic cells. It is responsible for modifying, sorting, and packaging proteins and lipids that have been synthesized in the endoplasmic reticulum for transport to their final destination. Now moving on to lysosomes, which is also known as digestive system of cell. The Golgi apparatus makes small structures enclosed in a membrane through a process called packaging. These structures are called lysosomal vesicles, and they are filled with hydrolytic enzymes that work best in an acidic environment. These enzymes can break down carbohydrates, proteins, lipids, and nucleic acids into smaller parts. That is why they are called as digestive system of cell. Like lysosomes, vacuoles are also membrane-bound organelles found in eukaryotic cells. However, unlike plant cells, animal cells lack well-defined vacuoles. It is a large, fluid-filled space surrounded by a single membrane called the tonoplast. The size and number of vacuoles in a cell can vary depending on the cell type and its stage of development. Vacuoles play a vital role in maintaining the structural integrity of the cell, regulating the water balance and ion concentration of the cell, storing nutrients, and waste products, and participating in cellular metabolism. In plant cells, vacuoles also have additional functions such as maintaining turgor pressure and storing pigments that give plants their colors. Moving on to another important organelle, this one is responsible for producing energy in the cell and is found in both animal and plant cells. It is our mitochondria. Mitochondria are tiny structures that cannot be seen easily under a microscope unless they are stained. The number of mitochondria in a cell varies depending on how active the cell is. They come in different shapes and sizes, but are usually sausage-shaped or cylindrical. Mitochondria have two membranes that divide them into two compartments, the outer compartment and the inner compartment, which is filled with a dense substance called the matrix. The inner membrane has many folds called cristae that increase the surface area of the organelle. Mitochondria are known as powerhouses of the cell because they produce energy in the form of ATP through aerobic respiration. They also have their own specific enzymes to carry out their functions. Mitochondria have their own DNA, RNA, and ribosomes for protein synthesis, and they reproduce through fission. Let's now focus our attention on another important organelle found in plant cells only known as plastids. Plastids are organelles found in all plant cells and euglenoids. They are visible under the microscope and contain specific pigments, which determine their colors. Plastids are classified into chloroplasts, chromoplasts, and leucoplasts based on their pigments. Chloroplasts contain chlorophyll and carotenoid pigments and are responsible for photosynthesis. Chromoplasts contain carotenoid pigments, which give plants yellow, orange, or red coloration. Leucoplasts are colorless and store nutrients such as carbohydrates, oils and fats, and proteins. Now, let's shift our focus to another organelle found in cells, which plays a vital role in protein synthesis. Ribosomes are a type of cell organelle that are found in both prokaryotic and eukaryotic cells. They are non-membrane bound organelles and are suspended in the cytoplasm. Interestingly, ribosomes can also be found inside two other organelles, the mitochondria and the rough endoplasmic reticulum, which we already discussed above. The rough ER is called rough because it has ribosomes attached to its surface. Ribosomes do not have any membrane surrounding them, which is why they are considered non-membrane bound. In plant cells, ribosomes are found suspended in the cytoplasm. Let's explore another important component of the cell that provides mechanical support and facilitates cell movement and maintains the cell's shape. The cytoskeleton is a network of protein structures made up of microtubules, microfilaments, and intermediate filaments found in the cytoplasm of cells. Now, let's see what helps our cells to move in fluid. Cilia and flagella are hair-like extensions of the cell membrane. Cilia move like oars to either move the cell or move fluid around the cell, while flagella are longer and responsible for cell movement. Prokaryotic bacteria also have flagella, but they are different from eukaryotic flagella in their structure. Let's see another organelle that helps in cell division. The centrosome is a tiny part of the cell that usually has two cylinder-shaped structures called centrioles, which plays a crucial role in the process of cell division. Now, let's see what are microbodies. Microbodies are tiny vesicles enclosed by membranes that contain different types of enzymes. These microbodies are present in both animal and plant cells. We hope that you have understood the topic of cell, the unit of life. Now, here is a secret on how you can study effectively with the EduRev app. You can learn with chapter notes, watch video lectures, and solve NCERT-based MCQ tests of this chapter on EduRev. And that's not all, you also get amazing courses for Class 11 and 12 biology, and much more for your NEET preparation. Thank you.